This protocol provides a way to assess the involvement of a certain factor in double fertilization of Arabidopsis. The fertilization status can be judged by the morphology of the sperm nuclei at a glance, so the quick obtaining of the data for statistical analysis is possible.
This method is specific to assessment for fertilization phenotype in Arabidopsis. There are still unknown fertilization regulators even if in Arabidopsis. This method would be useful for functional analysis of future unknown fertilization factors. To master the scale of quick and intact handling of the ovules are important for the success of this analysis.
To begin, grow Arabidopsis thaliana at 22 degrees Celsius under a 16-hour light and 8-hour dark cycle in a growth chamber. Cut and remove the first developed flower stalk with scissors to promote development of axillary buds. 2 to 3 weeks after cutting off the first stalk, measure the plant height. Plants with height above 25 centimeters are considered vigorously growing plants and are suitable for analysis. To emasculate, look for buds at stage 11 with bits of petals seen at the top. Use a number 5 forceps to remove sepal, petal, and stamen of flower buds. 15 to 18 hours after emasculation, take the stamen of a DMP9 knockdown line with background of HTR10 MRFP flower at stage 13 by pinching the filament with forceps. To pollinate, gently pat the stigma of an emasculated pistil several times with a